morning, everyone. Let me ring the bell. So good morning and welcome to Marine's Lebrec Thesis Defense, and thank you all for being here in person. I know you guys had to trek across the snow back in Santa Cruz, and thanks to all of our virtual attendees as well today. So my name is Max Grand, and I had the pleasure of working with Marine over the last couple of years here at Moss Landing. And before I leave the stage to Marine, I just wanted to give you a brief background and some more information about Marine. So, who is Marine? Well, I can tell you one thing. We share something in common. We have both deep, strong French roots. However, you will see later on today that our spoken English sounds actually much better than mine. In our free time, I think it's fair to say that Marine enjoys surfing, biking long distances, sampling various breweries in Santa Cruz, and she spends a lot of time in the summer back in France, in Osgore as well, which is a very nice place, and you should all go there at some point. I first met Marine in September 2019, when she visited Moss Landing and expressed interest in joining my lab. And soon after, her application package came, and it came with glowing reference letters, a very strong background in marine science, as well as very relevant professional experience, and you'll see that in a second. So the admission process was very, very easy for me. It was really a no-brainer. So Marine came with a Bachelor of Science in Oceanography from the University of Washington, which she got in 2016, and with that she came with a minor in marine biology, and a minor in Arctic Studies. I never heard about that before. And then after that, she decided to work for 2 years at UW at the Applied Physics Laboratory. And she worked there for about 2 years. And in 2018, she moved across the pond and landed in Monaco. And in Monaco, she worked at the International Atomic Energy Agency as an associate research scientist. And she worked with their Ocean Acidification International Coordination Center. And after all this, and lucky for me, she eventually made it to Moss Landing in the fall of 2020, in the middle of a pandemic. And first, she had to go through a full semester of virtual learning, which was not fun for them and for us also, instructors. But she picked things up very quickly. Already, she was in my lab doing experiments. I trained her, and it took about 3 days, and then she was on her way. So I hired her right away on my NSF grant as a graduate research assistant. And she wrote a very ambitious research proposal within a year, almost a year, after she started here at Moss Landing, which is very impressive for Moss Landing standard. And in that proposal, she said that she would first validate and optimize a new method for nutrient analysis in seawater. She said she would use it in various places, including our very own aquaculture facility. And she also said she would develop the first of its kind, open source, autonomous nutrient analyzer. And I was like, wow, great, take it away. But let me tell you something about Marine. When she says she's going to do something, she does it, and no matter what the challenge is. She made full use of our facilities, and here you can see her on the Boston Whaler sampling for nutrients in the Monterey Bay and Elkhorn Slough. You see her hen here at the aquaculture facility tasting some ulva, I guess, in the background. You see her on the Martin eating birthday cake after a long day trying our first prototype for underway applications. So she really, made full use of our facilities here. She became an oceanographer quite quickly and also a very skilled chemist, but Marine is very driven, and she knows that to make it in this field, you also have to have strong quantitative skills and strong programming skills. And with that, Tom Connolly hired her as a TA for his data analysis class back in 2022. She received 2 awards here at Moss Landing in 2022, the Moss Landing Scholar Award and the Coast Graduate Research Award as well. She presented her work at 2 major international conferences, including the Ocean Science Meeting last year, and this winter, she presented a poster at the American Geophysical Union meeting in Chicago. And she also took part in an oceanographic expedition this summer where she sailed on the Keel Moana, which you can see right here, to the Hawaii Ocean Time Series to test our instrument and validate it against standard methods. So clearly Marine is very skilled, and unfortunately for me, those skills were picked up very quickly. And I'm very pleased to say that in January, she started a new job at MBARI already as a data specialist. And there she manages and integrates all the data coming in from Senkus, and she also coordinates with various ocean observing networks. So the good news for me here, or I would say the only good news, is that she's not that far away from me. So now, before I leave you with Marine, I wanted to share a number with you. And that number is 89. So I'm thinking, of course, 89. That is the atomic number of actinium. And you're absolutely right. Of course, it is what it is, but that's not what I'm thinking right now. So let me explain, and I have a dirty little secret to tell you here. I have tracking devices on all the instruments in my lab. And these tracking devices track the total volume of fluids that are passed through the instruments. So of course, a couple of days ago, I checked on Marine's instruments, and I found out that since Marine started in fall 2020, she passed about 89 liters of fluids through her instrument. So you might be thinking, well, 89 liters, yeah, maybe I could drink that. Or you would think, you know, if we go back to our example of actinium, with 89 liters, you would be able to do only 3 samples, which is not that much. But what you have to remember here is that Marine developed a microfluidic method. And that microfluidic method consumes about 5 milliliters of fluids every single time Marine runs an experiment or a sample. So I've done the math for you because it's fairly complex math. And that means that since Marine started in fall 2020, she ran about 17,800 samples or experiments, which I think is fairly impressive. However, I forgot to mention something to Marine before today, is that the minimum requirements for a master's degree in chemical oceanography in my lab is 17,802 samples.
I'll be back.
Oops. So, and with that, I'll leave you with Marine, but I want you all to remember that developing a method in chemical oceanography is really difficult. It takes a lot of time, it takes a lot of effort, and it's mostly frustrating, sometimes rewarding, and it takes a lot of work to make some very tiny improvements, sometimes some tiny progress. And I must say that Marine has made much more than that, and I'm very proud.
Thanks, Max. Much, Max. That was such a nice introduction, and I promise I'm not going to go through each of those 17,000 samples today. I'll just get to the exciting part. So thank you all for being here today, those in person as well as those on Zoom. Really appreciate it. And so as Max alluded to, my thesis work is focused on developing new methods for measuring phosphate and silicate in seawater. Before I start, I just want to give a brief outline. So first I'll give a little bit of introduction and some historical context to this work, and then I'll jump into my thesis objectives, which are kind of threefold. So first, I wanted to optimize PFI and silicate assays on the bench, and then I wanted to adapt those methods for in situ operations. And then lastly, I wanted to actually apply those methods for coastal monitoring. And then I'll finish with some conclusions and some kind of lessons that I've learned along the way. And so to start off, I think it's important to kind of remind ourselves the important role that macronutrients play in controlling primary productivity in the oceans, which we already know. And so for my thesis, as I mentioned, I focused largely on phosphate and silicate, but I just want to mention that nitrate and nitrite are also important macronutrients. And so dissolved inorganic phosphorus in the form of orthophosphate is really important for various things in the ocean, including the formation of DNA and RNA, as well as forming cellular structures and through various metabolic pathways as well, like fixing organic carbon via photosynthesis. Silicate, which is important, or which is found in the form of silicic acid in its dissolved state, is really important for the formation of diatom tests or shell walls. And as many of you know, these diatoms are the most abundant type of phytoplankton in the ocean, making this nutrient really relevant. And this is all quite relevant when we think about energy transfer through trophic food webs, and also in terms of carbon fixation and long-term burial of that carbon in deep sea sediment. And so imbalances in nutrient stoichiometry can be a really important indicator of ecosystem health and water quality. And this is particularly relevant in coastal regions where anthropogenic processes come into play. And so, for instance, phosphate anthropogenic loading can occur from things like agriculture fertilizer, wastewater, industrial waste, among other things. And that eutrophication can lead to kind of detrimental impacts like hypoxic or anoxic events, as well as higher prevalence of harmful algal blooms. In contrast to that, silicate depletion can be an issue, particularly due to things like river damming and urban development. And we've seen this actually play out, this idea of nutrient stoichiometry affecting water quality or ecosystem health. So here in our own backyard of Monterey Bay, during the 2015 marine heat wave event, there was a really large harmful algal bloom, which some of you probably know. And Ryan et al. Hypothesized that the reason those pseudonychia were so toxic and able to thrive so well was because of a silicate depletion relative to nitrate. And so that brings me to my next topic, which is, well, how do oceanographers actually measure nutrients in the ocean? And so for the last several decades, we've largely relied on these kind of large-scale, basin-scale hydrographic surveys through programs like GOSHIP and the World Ocean Circulation Experiment, just to name a few. And these are really important programs because they allow us to look at nutrient dynamics changing over long time scales, like decadal scales, and also to look at nutrient distributions across basins. But to look at nutrient dynamics happening in coastal regions, we need different monitoring approaches. And so one of the, I would say, challenges with coastal nutrient monitoring is that many programs still largely rely on someone actually physically going out in the field and collecting discrete samples. And this is really useful, but ultimately we need higher resolution sampling to better understand what's happening in these systems. And so to give a bit of an example, these are nitrate concentrations measured here at the MLML shore station throughout the month of January. And these concentrations were measured using a UV nitrate sensor. And so these are the concentrations that you would see if you only went out and collected one sample a week for a month. And so we can start to see that there's a high nitrate event happening towards the end of the month, but we really don't know much else. If we change our sampling frequency to daily, then we start to see kind of the exact timing of that event, and we start to see some diurnal variability in concentrations that we otherwise wouldn't pick up. If we then change our sampling frequency to hourly, then we get to see a lot more dynamics of actually what's going on in these systems that might be driving that variability. And with that high sampling frequency, we're also able to capture these two high nitrate pulses that we otherwise would have completely missed. And so as oceanographers, we can measure nitrate really well using these UV nitrate sensors, but those capabilities just don't exist for phosphate or silicate. And so to really bring this point home, high resolution nutrient monitoring is really valuable in dynamic systems, but we really just can't do that as it stands for phosphate and silicate. And so since my thesis is mainly focused on method development, I thought that it'd be useful here to offer a bit of historical context. And so really prior to really the 1960s, chemists were running chemical assays by manually mixing solutions together and putting them in a detector. And it wasn't until 1957 that Leonard Skaggs invented what's referred to as segmented flow analysis. And this was quite revolutionary because it allows much more samples to be run, and you can run several analytes on the same machine itself. And it's actually still considered to be the gold standard today. But over time, there's been a lot of work done to kind of miniaturize these instruments. And so in 1990, Yarda Ruzicka invented what's referred to as sequential injection analysis. And this is really great because it's miniaturized, so it uses a lot less reagents and samples. And it's also a lot easier to operate and requires less maintenance. But at the same time, it's still not optimal in terms of mixing efficiency. And so that's where programmable flow injection comes in, which from now on, I'm just going to refer to it as PFI because it's easier. And so in 2018, Yarda Ruzicka, again, invented PFI. And this is really exciting because first off, it's fully miniaturized. So as Max alluded to, it uses really small reagent and sample volumes. It also uses really efficient mixing, which I'll talk about a little bit later. And one of the exciting things is that all of the different components of the instrument can be controlled using a computer. That means that if I build a script for my assay that I'm using, I can share that with someone else and they can reproduce exactly what I've done. And lastly, these instruments are also more affordable relative to kind of standard methods, which makes them a little better for wider use. So let's look at PFI a little bit more closely. Here you're seeing images of the front view and the top view of the instrument. And I'll just point out kind of the main components of it. So what you're seeing is there's an 8-port valve that rotates, and each of the different ports go to different parts of the instrument, whether that be your sample, your reagents, a waistline, or the flow cell. Then there's those 2 milligat piston pumps, which work bidirectionally, so they can either aspirate or dispense solutions. And then those 2 holding coils are temperature controlled, and that's actually where the mixing of the assay takes place. The detection system is pretty straightforward. It has, in this case, a 20-centimeter flow cell. On one side of it is a light source, and on the other side is a spectrophotometer. So diving a little bit into the chemistry, how can I actually use PFI to measure phosphate and silicate? So for my thesis, I'm using classic molybdenum blue chemistry. The chemistry is pretty similar for phosphate and silicate, so I'll just walk you through the phosphate method. And so for phosphate, you have phosphate ions reacting with acidified molybdate to form phosphomolybdic acid. That acid is then reduced, in this case using ascorbic acid, to then form a blue compound that's referred to as phosphomolybdenum blue, or in the case of silicate, it's silicomolybdenum blue. And so I'll just walk through each of the mixing steps so that you have a better idea of actually what's going on here. So the first step is to aspirate my sample into one of the holding coils. And then the next step is to aspirate my acidified molybdate into another holding coil at the same time as my sample is being dispensed into that holding coil. So you essentially have both of the solutions merging together, and as it's doing that, it's mixing efficiently. The third step is to then aspirate my ascorbic acid into a holding coil while the molybdate and sample solution is dispensed into that holding coil, where that reduction step happens. And then once all of the different reagents and sample are mixed together and the reaction takes place, then we can send the reacted product to the flow cell to be detected. And so that's what you're seeing here is an absorbance spectrum with absorbance on the y-axis and the wavelengths on the x-axis for both phosphate and silicate spectra. And using a particular wavelength where the phosphomolybdenum blue or silicomolybdenum blue peaks, we can monitor the absorbance at that particular wavelength. Then using a calibration curve with standards of known concentrations, we can measure the concentration of our sample, which is what we're trying to find out. So that brings me to my first thesis objective, which was to optimize and validate methods for analyzing phosphate and silicate using PFI. And the rationale here is that I'm definitely not the first one to develop these methods. There has been work done to develop these methods in the past, but I wanted to kind of further optimize these methods. And to do that, my objectives were to increase the sensitivity and the precision of these methods, and I also wanted to minimize matrix interference, which I'll define a little bit in my next slide. And then lastly, I wanted to validate these methods against established methods that are already being widely used by the community and against certified reference materials. And so arguably one of the most important components of molybdenum blue chemistry are the concentrations of that molybdate and of the acidity of the solution. And so specifically, if the acidity is too high relative to the molybdate, that'll result in a complete degradation of the product. But on the other hand, if the acidity is too low relative to the molybdate, then the molybdate itself can be directly reduced, and that leads to a high reagent blank, which is a problem. And so there have been previous studies that have specifically looked at optimizing the molybdate and H+ concentrations, specifically with PFI. So this here is a figure by Ruzicka et al. In 2019. On the y-axis is the calibration slope, and on the x-axis is the acidity, and the different colors are the different molybdate concentrations. And so what you can clearly see is that if the acidity is too high or too low, you see a complete drop in sensitivity. And these are really insightful results, but one of the things that's kind of lacking from these kinds of studies is that folks have only really looked at the effect of molybdate and H+ on sensitivity, but none have really looked at the effect that it might have on matrix interference. And by that I mean, if I calibrate my instrument in millicu water, but my sample is in seawater, do I get a difference in signal between the two? And if so, that's going to affect the accuracy of my method. And I'll just mention here as well that calibrating your instrument in millicu water is much more advantageous. Most research labs will calibrate their instrument in low nutrient seawater, but as you can expect, it's much harder to actually access low nutrient seawater, whereas most labs around the world have access to millicu water. So to do this optimization work, I changed the concentrations of my molybdate and H+ at the ranges that you're seeing there, and I did this for both silicate and phosphate. So to test the analytical sensitivity, I simply ran calibration curves for each of those treatments. And then I also wanted to test the matrix interference, and to do that, I ran spike and recovery analysis. If you're unfamiliar with spike recoveries, what I'm essentially doing is just running a calibration curve with standards that are prepared in millicu water. Then I'm spiking a known concentration of my analyte, whether that be phosphate or silicate, into a low nutrient seawater matrix. I then will analyze that spiked sample using PFI. And since I know the known concentration of my spiked sample and the concentration that I measured, I can calculate the % error of my method. To give a quick example of that, say I spike my low nutrient seawater with 2 micromolar phosphate, and then I analyze that same sample and measure 1.9 micromolar, then I can get a 5% error, which is pretty good. So to dive into my results, first I'll show the silicate results, and then I'll show you the phosphate ones afterwards. What you're seeing here on the y-axis is the calibration slope, and on the x-axis is the acidity at 3 different molybdate concentrations. And so here what you can clearly see, similar to that Ruzicka paper that I showed earlier, is that there's kind of a sweet spot where the sensitivity is maximized, and if the acidity is too high or too low, there's a complete drop in sensitivity. Another interesting thing that I observed is that if the molybdate concentration is higher, there tends to be higher variability in my slopes, which corresponds to those bigger 95% confidence intervals that you see. If I also include the % error of my spike recoveries, which again is, in this case, kind of a proxy for matrix interference, you can see that I was able to minimize matrix interference at the same acidities that maximized the analytical sensitivity. And this is pretty cool because this is the first time that this has been shown really for silicate. The phosphate results are a little bit more messy, so I'm not going to discuss them really, but I just want to mention that the takeaway here is that if you increase the molybdate concentration by too much, I was never able to observe % errors in my spike recoveries that were less than 5%. So I find it actually more useful to use lower concentrations of molybdate and an acidity of around 0.1 molar. So next I wanted to see, well, how do these results actually compare with the literature? So to do this, I did a literature review of papers which report their molybdate and H+ concentrations, and which also report whether or not they have matrix effects or report their matrix effects. So what you see on the y-axis is the molybdate concentrations that were reported by these papers, and on the x-axis is their acidity. And so as you can see, there was only one paper that didn't report, or that reported not having any matrix effects. And interestingly, my optimized phosphate method, where matrix effects were minimized, lies really close to that Deng et al. Paper, which is pretty cool. That other green dot is the optimized silicate molybdate and H+ concentrations. And what I noticed here is that other silicate methods that I use, or that I found, use higher molybdate concentrations, which might be accounting for their reported matrix effects. So the takeaway here is that, based on my experiments, is that if the molybdate and H+ concentrations are carefully optimized, matrix interference may be eliminated, and that's largely been overlooked, I think. So now I'm going to switch gears a little bit and talk about ways in which I optimized PFI mixing volumes. And I specifically wanted to do this for the phosphate assay. So more specifically, phosphate concentrations in the ocean are quite low, reaching concentrations of about 3 micromolar, whereas silicate can get as high as 180 micromolar. So it's pretty relevant to try to increase the sensitivity of the phosphate assay. So one way to do that is to modify the volume of sample relative to the reagent. And so by that I mean when I mix my sample and reagent together, the volumes of each of them is going to dictate how much of that phosphomolybdenum blue is actually formed. And what we can clearly see here is that, so here on the y-axis is the calibration slope, and on the x-axis is the different sample to reagent ratios that I tested. And yeah, what we can see here is that as you increase that sample to reagent ratio, as expected, I also see an increase in calibration slope. As I increase the sample to reagent ratio, I can also decrease my limit of detection, which is essentially how low of a concentration my method can measure accurately with high confidence. Ultimately, I ended up using the 6:1 sample to reagent ratio because I found 60 nanomolar to be a really good LOD, and it was just also more practical to use. And so using the optimized methods that I just described, I also ran certified reference materials, which are samples of known concentrations. And this is really useful for validating the accuracy of my method. So there's a lot of numbers and a lot going on in this slide, but I just want to mention on the top is the phosphate and on the bottom is silicate. And the first column of the table is the CRM concentration, which is the true value. And then the next column is the calculated concentration that I calculated myself. And the takeaway here is that for both of these assays, the calculated concentrations that I measured lied within 5% of the true value. So my next step here was to validate these methods against state-of-the-art analytical techniques that are already being widely used by oceanographers. So to do this, I was really fortunate to go on a research cruise up to Station Aloha as part of the Hawaii Ocean Time Series program last August. And so during this cruise, we took discrete samples from Niskin bottles from the surface to about 4,800 meters. And at each of these depths, we collected replicates. And we did that so that one of the replicates could be sent to Scripps Institute of Oceanography to be measured using an autoanalyzer, which uses that segmented flow analysis that I mentioned earlier. And then the other replicates were measured by myself using the PFI that I brought with me to Hawaii. So what you're seeing here are the nutrient profiles from the cruise that I analyzed using PFI. So on the left is phosphate and on the right is silicate. And the y-axis is the depth, and on the x-axis is the concentration. These are pretty typical nutrient profiles in the open ocean, where you see low concentrations near the surface due to phytoplankton uptake, with a general increase with depth due to sinking organic matter and remineralization of that organic matter back to its inorganic form. And so overlaid here are the concentrations that were measured by the Scripps lab. And what we can see is that both profiles overlay each other quite nicely. So I wanted to look at this a little bit more quantitatively. So to do that, I ran linear regressions between the SIO measurements and my own. So what you're seeing on the left-hand side are those linear regressions with the SIO measurements on the x-axis and the PFI measurements on the y-axis. On the right-hand side, you're just seeing the difference in concentration between the two methods as a function of depth. And so the negative values correspond to underestimations that I measured relative to Scripps, and the positive values are overestimations. And what I found here is that there was a significant difference between the SIO measurements and my own for phosphate. But when we look at the difference in concentrations, the difference never exceeded much more than about 100 nanomolar, or 0.1 micromolar, which is pretty negligible. For the silicate samples, there was no significant difference between both methods, despite some variability that I observed, particularly at deeper depths, where I found my measurements to be slightly underestimated relative to SIO measurements. Overall, though, we can say with pretty high confidence that these optimized benchtop methods that I used can produce measurements that are consistent with state-of-the-art methods that oceanographers are using. Okay, so this brings me to my second thesis objective, which was to adapt benchtop methods to be able to use PFI in the field autonomously for extended periods of time. And the rationale here is that these benchtop methods that I just described to you are really useful and great, but there's several considerations or adaptations that need to be made before you can leave an instrument out in the field for long periods of time. So specifically, I wanted to look at the long-term stability of reagents and standards to see if I can actually leave my instrument in the field for, say, a month. I also needed to figure out how can I actually get the instrument to calibrate itself while it's in the field. So when you're in the lab, you can calibrate your instrument manually whenever you want, but once it's in the field, you have to figure out how is this instrument going to be calibrated. And then lastly, I wanted to run a pilot deployment just to make sure that this instrument could actually function in the field. So as I mentioned, the first question here that I wanted to address was how long can my reagents and standards remain stable at a range of temperatures? And so each of the reagent and standard solutions that I use kind of come with their own set of challenges when it comes to stability. So for instance, the ascorbic acid reagent tends to degrade when it reacts with oxygen or when it reacts with light due to photodegradation. And so that's an image of what ascorbic acid looks like after several weeks or months once it's fully degraded. And so to try to get around this, I prepared several different treatments, including trying to modify the concentration of the ascorbic acid. And I also tried purging it with nitrogen gas to essentially remove any oxygen from solution to avoid oxidation from happening. I also tried spiking it with acid, and I tried modifying the surfactant in the solution. I also found that the molybdate used for the silicate assay tends to precipitate after several days to weeks, which is an issue. And so to try to avoid that precipitation from happening, I tried modifying the acidity and the molybdate concentration to try to see if that could be avoided. And that's an image of what the molybdate looks like once it's precipitated. And then lastly, standards can also be compromised if they adsorb to storage walls of the container that they're in, or also due to consumption by microorganisms. So if your solution isn't sterile and there's microorganisms in your standard solution, that could consume the nutrients and therefore degrade that sample. And also speciation can be an issue, particularly for silicate. And so to try to get around this, I wanted to see if spiking it with acid might reduce that degradation from happening. So to run this experiment, on day 0 of the experiment, I prepared fresh standard and reagent solutions. And with those solutions, I ran calibrations for each treatment. And then I stored them in either a temperature-controlled room or a water bath. And then I recorded the temperature using a hobo temp logger. Throughout the experiment, I produced 3-point calibrations every couple days. And if the sensitivity of those treatments dropped by more than 10%, I prepared new standard or reagent solutions successively until that sensitivity went back up. And then I continued this experiment for at least 30 days because that was kind of my initial goal, to try to keep these solutions stable for that long. And I continued that experiment either until the solutions were completely used up, or if the sensitivity was completely lost, then there was really no point in continuing. So diving into some of my results here, these are my results showing the stability of ascorbic acid for each treatment. So on the x-axis is time, and on the y-axis is the calibration ratio. And it's a ratio because I wanted to compare my calibrations relative to day 0. And the different colors are the different treatments that I ran. And what we can see here is that despite that day-to-day variability that I observed, all of the treatments were quite stable in that they remained within 10% of baseline levels, which was my kind of goal. I then tested the control treatment where the ascorbic acid wasn't modified because I found that one to be quite stable, and I tested it at a range of temperatures. And that range of temperatures is consistent with generally temperatures that are observed here in Moss Landing. And what I was able to find, again, is that regardless of the temperature, those treatments stayed within 10% of baseline levels. Moving on to the stability of the standards, here the lines with the X's on them are standards that have been unacidified, and the ones with the dots are ones that have been acidified. And here on the y-axis is, again, it's a ratio, but it's relative to, day 0, and in this case, for a 1 micromolar phosphate standard. And here what we can clearly see is that if the standards are not acidified, they degrade much more rapidly than if they are acidified. And the ones that are acidified remain close to 10% of baseline levels for over 30 days. Moving on to the stability of the molybdate reagent. So here I found that by decreasing the molybdate concentration, I was able to avoid that molybdate from precipitating. And going back to my earlier experiments, I had found that using a 90 millimolar sulfuric acid concentration not only avoids precipitation, but it also falls where matrix effects are minimized. So then using that treatment, which is 4 millimolar molybdate and 90 millimolar sulfuric acid, I wanted to test the stability of that over a range of temperatures again. So what you're seeing on the x-axis there are the different temperatures that I tested. And the light blue is the start of the experiment. The darker color is the end of the experiment. And again, we can see that I was able to remain within 10% of baseline levels, meaning that I was able to find molybdate composition for that silicate assay that remains quite stable. And so the takeaway throughout all of these experiments is that I was able to find standard and reagent solutions for both phosphate and silicate that can be kept stable for 30 days. Okay, so now I'm going to shift gears a little bit and talk about calibrating the instrument while it's in the field. And so there's kind of several different approaches that could be taken to calibrate your instrument once it's in the field. You can either use a single standard, and so run a blank and a single standard, but that's not ideal because it doesn't give you any information about the linearity of the calibration. Another approach is to use multiple standards that are hooked up to the instrument, but that's also not ideal because I'm limited by those 8 ports that I mentioned earlier. So one way to get around that is to produce a multi-point calibration using a single standard that's diluted online. What I mean by that is, say I have a 3 micromolar phosphate standard. If I dilute that standard using my milliQ carrier, I can produce a working standard within the instrument. So here, if I aspirate 600 microliters of my standard, at the same time as I dispense 300 microliters of milliQ water, I can produce a 1.5 micromolar phosphate standard within the instrument without me ever having to manually do it. So after I developed this calibration protocol, I wanted to make sure that I can achieve the same level of precision and linearity as if I was manually preparing those standards. So that's what you're seeing here are the 2 different calibration curves using manually prepared standards and online dilution calibrations. And we can see that they're very similar to one another. So what that means is that the auto dilution protocol that I implemented is able to achieve very accurate mixing. So the last step for this kind of objective of my thesis was to deploy the instrument for several days as part of a kind of pilot deployment using that self-calibration protocol that I just mentioned. So to do that, I left my PFI instrument for 5 days at the MLML pump house, which has a seawater intake pipe at around 14 meters depth near the head of the submarine canyon. And auto dilution calibrations were run every 12 hours throughout the deployment, and samples were collected every hour. So here you're seeing on the very bottom panel is the phosphate concentrations that I measured throughout that period, and the other panels are other hydrographic parameters that are measured at that same shore station. So here you're seeing also nitrate, dissolved oxygen, temperature, and tidal height. And I included those vertical lines just to show that those spikes in phosphate concentrations that I measured coincide pretty nicely with other spikes in the other parameters that were measured at the same time. Okay, so that brings me to my last thesis objective, which was to implement field-based PFI deployments for various coastal monitoring applications. And so the rationale for this is that PFI has been shown to be a really useful benchtop tool, but it really hasn't been shown or actually implemented in the field for in situ monitoring. And so for this work, I, with a team, developed an open source PFI prototype for monitoring at coastal shore stations. And then lastly, I want to finish by showcasing a few ways in which PFI was used for various applications. So with a team at San Jose State University, we developed this field deployable PFI prototype named Fiona, which is the PFI Ocean Nutrient Analyzer. And this instrument is really exciting because it uses commercially available hardware and open source software that we developed. And so here you're seeing all the different components of the instrument with the electronic components as well. It uses a Raspberry Pi microcomputer to send commands to the communication board, which then controls the different hardware components. I've also included here, which is kind of difficult to see, but it's a real-time logging system, so you can, as a user, be able to see what the instrument is doing exactly in real time, and you can access that information remotely. So this was a really cool tool that we developed and I thought was really useful in terms of troubleshooting. So after this was developed, my main objective was to figure out, well, could this prototype actually be used to collect some high-resolution measurements for about a month? So to do this, I had the Fiona instrument deployed at the MLML shore station for 1 month for each assay. So first I did a phosphate deployment, and then I did a silicate deployment after that. And here you can see an image of Fiona at the shore station above all the other hydrographic parameters. If you've been down there, you know what that looks like. And so during the deployment, samples were collected using an auxiliary sampling pump, and the instrument was calibrated every 3 hours. It was calibrated by just running 2 blanks and then triplicate standards. I could have implemented that auto dilution protocol that I mentioned earlier, but it was a little bit too complex to program, but I think it could certainly be done in the future. Another consideration that I had to make here was that was to increase the incubation period in the flow cell before the absorbance is actually monitored. And so specifically, I found that using 5 minutes was an adequate amount of time until that absorbance signal stabilized. And this was important because those holding coils for Fiona are not temperature controlled, meaning that the reaction that's taking place is a lot more dependent on the ambient air temperature. So first, I'll show you guys the PFI phosphate deployment, which happened between October and November of last year. And first, I'm just showing you the calibration information with, on the top, you're seeing the absorbance of blank samples over time, and on the bottom is the absorbance of my 2 micromolar phosphate standard. Interestingly, the standard signal declined, which was surprising because the standard was acidified, which kind of goes against those, that experiment that I showed earlier. But ultimately, that doesn't really matter because I was calibrating my instrument every 3 hours, which was accounting for many changes in sensitivity. So during that period, 678 phosphate samples were analyzed. What you're seeing here on the top is my phosphate time series from the deployment. And then again, similar to earlier, I'm also showing all the other some other hydrographic properties that are measured there. And so phosphate concentrations ranged from 0.2 to 2.4 micromolar, which suggests that there's a lot of natural variability in the system, which I'll talk about a little bit later on. And I just want to mention here that there are some gaps in the data, and I just also want to mention that, yeah, the deployment was not completely continuous, so there was a moment where one of the pumps malfunctioned, one of the lamps fried, there was a clogging issue, so it wasn't 100% perfect, but that's how it goes. So this is now the Fiona silicate deployment, which happened between November and December of last year. And one interesting observation here is that the standard signal fluctuated throughout each day, as you can see there, which, as we can see, is pretty strongly correlated to the ambient air temperature. And I think this is due to the fact that the silicomolybdic acid formation is quite temperature dependent. And so even though I had that 5-minute waiting period at the end of my mixing, there's another mixing step when the sample and the molybdate mix that wasn't accounted for. But again, because I was calibrating my instruments so frequently, that those calibrations were accounting for those changes in sensitivity of my silicate standard. So during the deployment, almost 500 silicate samples were analyzed. Again, you're seeing silicate on the top and the other parameters on the bottom. And concentrations ranged from about 4 to 29 micromolar. And then I'll mention here as well, there were a few instances where the software itself was having issues and the instrument had to be rebooted manually. So that's why you see those gaps there. And I just want to show here, if we actually zoom in, because when it's zoomed out, it's a little bit hard to see. But if we zoom into the first week of the deployment, we can see that the spikes in concentrations correspond quite nicely with other parameters. And here I've just showed salinity because it has quite a strong signal. And so to look a little more closely at how nutrient concentrations vary with other variables measured at the shore station, I ran linear regressions between each nutrient and other key parameters, including temperature, salinity, pH, and dissolved oxygen. And what I observed is that phosphate samples, which are higher in concentration, tend to be colder and also more saline and have lower pH and lower dissolved oxygen. And similar trends were also observed during the silicate deployment. And so this high nitrate cold water signal is indicative of deep water that tends to have lower oxygen and pH and also tends to be colder. And this is due to internal tidal waves that propagate deep water up the canyon shelf into the nearshore environment. And this signal is seen at semi-diurnal tidal cycles. And so that's why we see that trend there. And I just want to also include these TS plots here, which kind of conclude the same thing, which is that high phosphate samples were observed in seawater samples, which are high in salinity and low in temperature. And of course, there's some variability in the system. So for instance, the internal tidal cycle tends to vary over time. And there's also probably other factors that influence water, the composition of the water, such as precipitation events potentially, runoff from the Old Salinas River, wind mixing events, and other factors that might influence that. To summarize, though, I think it's valid to say that these deployments really kind of served as validation that PFI can be used for field monitoring to collect some pretty valuable information about nutrient dynamics on short timescales. And so as with any kind of first attempt at a deployment, it didn't come without any kind of challenges and lessons learned along the way. And so specifically during both deployments, something that I observed was a noticeable amount of carryover. And so by that I mean when a sample was run, it was likely influenced by the sample preceding it. And so for instance, to calibrate my instrument, like I mentioned earlier, I was running triplicate standards. And consistently, the first standard that I ran tended to have a lower signal than the second and third triplicate. And that was probably because it was influenced by the blank sample, which is just millicue water that was run before it. Some other issues that I mentioned earlier, which I'll just touch on, are the light frying issue, the temperature effects on silicate sensitivity, which likely affected my accuracy a little bit, and also some inconsistent pressure in the sample line itself that probably caused some back pressure in the system. Okay, so I want to finish my talk by giving kind of one last example of a way in which PFI was used in the field. So specifically, I used it for measuring phosphate removal from macroalgae at a seaweed aquaculture farm, at the one just down the street. To give a little bit of background, seaweed are very efficient at removing pollutants and nutrients from seawater, and they've been used for various bioremediation strategies. And there have been a lot of studies aimed at quantifying nutrient removal efficiency from various species, but many of these studies are aimed at looking at nutrient removal on kind of long timescales, like days to weeks, and there really aren't a lot of studies looking at those nutrient removal dynamics on shorter timescales. And then I'll also mention that ulva is a green algae, which is very efficient at removing pollutants and nutrients from seawater, in large part due to its high surface area to volume ratio, and it's also quite resilient in stressful environments. So my main objective here was to use PFI to estimate phosphate uptake rates of ulva specifically at the MLML aquaculture facility. So to do this, I used 3 different treatments, including one treatment where I didn't have any ulva in the tanks, and my 2 other treatments had different stocking densities. I placed the specimen in these large 370-liter aquaculture tanks, and I added a stream of bubbles to keep the algae suspended to replicate tumble culture, which is how these specimens are typically grown in aquaculture. The tanks themselves were spiked to reach concentrations of about 2 micromolar, and that was really just to be able to see that signal in drawdown as the seaweed were removing it. And then every 5 minutes for a period of 4 hours, new samples were being analyzed from the tank so that I could see that change in concentration over time. And then after the experiments, I also calculated the phosphate removal efficiency, which is just a function of the beginning and ending concentrations. And then I also wanted to calculate the specific uptake rate, specifically normalized to the fresh weight of the algae. And so what I found is that for both ulva treatments, they removed between 83 and 100% of the phosphate present after the 4-hour period, which is pretty cool. And I'll also add that the control treatment saw virtually no change in phosphate concentrations between the beginning and the end of the period. And so what I'm showing here is the weight normalized phosphate uptake rate as a function of phosphate concentration. This data is a little hard to interpret, but really the main takeaway is that the uptake rates for both treatments were not statistically significantly different, meaning that increasing the stocking density didn't necessarily increase the uptake rate. My hypothesis for that is that if you have too much algae in a tank, that can lead to self-shading, and that could mean that certain specimens were unable to photosynthesize or assimilate nutrients as efficiently. But regardless of that, I think the takeaway is that here is that PFI can serve as a pretty useful tool to sample and quantify phosphate removal rates and could be used for other similar kinds of applications. So that was a lot, so I'm just gonna summarize kind of my main achievements here. So first, I was able to optimize PFI benchtop methods for both phosphate and silicate, and I did that by increasing not only the sensitivity, but also minimizing matrix interference. I was then able to validate these methods against CRMs and also against widely used methods. I then adapted those methods to be able to use PFI in the field. And then with a team, we developed a field deployable open source PFI prototype. And then lastly, I was able to use PFI for high resolution sampling for some new pretty cool applications. Sweet, well with that, I really just want to thank everyone who helped me get here. So first I want to thank my awesome thesis committee, Max, as well as Tom Connolly and Mike Graham. Thank you guys so much for all of the support throughout the last couple of years. I also want to thank my funding sources, including the NSF, which funded this project, and I also want to acknowledge San Jose State, as well as some scholarships that I received through COAST and MLML. I also want to thank the entire San Jose State engineering and computer science team. This is an image of some of the students that worked on that project after they finally got it to work, so I just want to thank them all for their expertise and knowledge. And then I also want to thank the University of Hawaii Manoa Chemical Oceanography Lab, including Mariko, Chris, Yarda, and Maddie. That's a photo of Maddie and I on the hot cruise, very sleep deprived on the last day. And then I just want to thank the entire MLML community. Big shout out to the Chemosh Lab, Seamus and Carol, who recently joined our lab, as well as Eric and Jess. Just want to thank you guys. And I just want to thank also the whole MLML faculty and staff, including Tara. I also want to give a big shout out to Jocelyn, as well as IT and the library staff. And I also want to just acknowledge Steve and Madi, who helped me a lot with those field deployments. They're some of the technicians that I've worked with. And then I also want to thank the shop team, the ship ops team, and the diving program, which was definitely one of my favorite parts of being part of a student here. So, and definitely last but not least, I want to thank my awesome family and my amazing partner Mal for all of their support. And thank you so much for your time.
Thank you all. We have time for questions, and for those of you guys on Zoom, the audio system is not working so well, so if you have questions, please put them on the chat, and I will ask those questions to Marine, and you'll hear the answer. So feel free to ask your questions there as well.
Yeah, Diana.
I have a question about the Hawaii data and the comparison to the scripts data.
Yeah.
And because they're offset in different directions, I was wondering what, and that was related to death, I think. What do you think that was from? And could it be related to the time of citizen analysis?
Yeah, that's something that I've thought a lot about, and actually, especially for silicate, I know that other studies have looked specifically at this, where so we froze the samples when we sent them to scripts, and so something that I thought a little bit about is, does that change in concentration that I observed, was that due to inaccuracy on my side, or is it due to preservation of the samples? And it's really hard to know. I think another thing that I'll mention is that that offset seems bigger at deeper depths because the concentrations are bigger at deeper depths, even though the percent difference might actually be only a couple percent. And so I think it's kind of all relative in that way. So yeah, good question though. Yeah, Annie. Oh, yeah.
Marnie, that was awesome.
Thank you.
I can't believe how many ideas you had that worked out so successfully. I was like, was there anything that you did that didn't improve and optimize this method? It didn't seem like it. Just to ask a specific one, when you were adding HCl, I think in the stability optimization, what motivated you to do that? And do you think that that tackled all of those kind of bullet point challenges you had, or do you think one was really causing more of the issues?
Totally. Yeah, I think in terms of standard preservation, there's a lot of different methods that are out there for trying to keep standards stable. Some folks will use like mercuric chloride or chloroform, but something that I saw in the literature that was also used is just spiking it with acid. And I think it's also less, well, I mean, working with mercuric chloride or chloroform is not ideal. So that was kind of my first go at it, is let's see if spiking it with a little bit of acid might kill off any microorganisms that might be in there. So potentially didn't solve that absorption or some of the other issues that might occur. And that's something that I was also wondering is, like, during my Fiona deployment, I did see a little bit of decline in my standard sensitivity. And I'm not really sure why, because I did spike it with acid, the same concentrations that I used for those stability experiments. So there might be something else going on, like temperature fluctuating a lot more or something in the field that I just didn't account for, but yeah. Thanks. Yeah, Jocelyn.
That was amazing. The depth and breadth of your study was phenomenal. Did you optimize the reaction time on the benchtop similarly to what you did for the flow-through stuff, or was it about the same time frame?
I did, yeah, thanks for asking that. Actually, so yeah, one thing that I'll mention is that for the benchtop method, those temperature, or those holding coils are temperature controlled, so the reactions happen a lot faster. So I did optimize that for the benchtop, but typically I wasn't waiting more than 1 minute in the flow cell. So that makes the sample throughput a lot higher because I can run a lot more samples just because it's a faster process. But for Fiona, I had to change it to 5 minutes just to be able to see that the absorbance actually stabilize. And I mean, even still, 5 minutes is not too bad, especially since my goal was to get hourly measurements. Taking 5 minutes doesn't matter because for the next 55 minutes, the instrument is just sitting there anyways. So, yeah. Yeah, Mike.
So your small data set's probably one of the first ever for showing seaweed uptake in real time, which is pretty cool, right? And that's mostly because a lot of groups focus on nitrate, or nitrogen, and there hasn't been much work with phosphate, even though in fresh water and in certain areas like the Gulf of Mexico it's really important. just thinking broadly, does this carry over to freshwater systems easily, or is there already an analogous system set up? Because, you know, the application here is great, but there's other places around the planet where I can see this being very desired for those particular anthropogenic impacts.
Yeah, and I mean, like, as you said, here, especially in Monterey Bay, we don't have, it's not a very eutrophic system. I mean, yeah, the Elkhorn Slough is, but the ocean itself is not really. And actually, the ambient seawater phosphate concentrations were like 0.4 micromolar, so I had to spike it with a lot of phosphate to actually see that signal. I have seen some other papers using a similar type of approach, specifically using algae as bioremediation tools for like fish pond effluent in freshwater systems specifically. So I think, but you know, even still, there aren't a lot of studies looking at that drawdown happening in real time. So certainly a lot of applications potentially with fish farming and other systems that might be a lot more eutrophic. So, and yeah, for this work, I specifically focused on seawater. This is a marine science program, but you could certainly use it in freshwater, I think, too. So. Yeah.
So you showed a really nice application of this technology to the internal tidal waves. What would be the next oceanographic process you think this technology would be really good to target and answer some questions?
I think it'd be really cool to see this, well, to have something like Fiona deployed for a longer period of time would allow us to look at things like upwelling signals, especially, you know, we have the nitrate sensor there, but what about phosphate and silicate? We don't really have a good understanding of how upwelling is affecting those nutrients, especially in the near shore here. And so if we had kind of a suite of sensors, of nutrient sensors, you know, even here at the Moss Landing Shore Station, we would get a lot more information about all of these macronutrients and how they might be changing over time. So, you know, that one month time period that I had the deployments were useful, but it really didn't give me any information about any seasonal variability that might be going on. Yeah, Jocelyn.
Did silicate interfere with phosphate or vice versa, or were they different enough in their spectrophotometric?
It's actually the other way around. Phosphate tends to interfere with silicate, and so one way to get around that is to use oxalic acid. So I didn't mention it in this talk, but yeah, one of the other reagents that I use for the silicate assay is oxalic acid, and that essentially destroys that phospholipid blue complex and completely avoids that interference. Yeah, but another thing that I'll mention is that the acidities of both phosphate and silicate assays are quite different, and so because of that, I think that also might remove some of the interference, because that was never really an issue for me.
In the beginning, you showed the Ryan and Ray Pudella HAB paper. Do you anticipate a harmful algal bloom detection or early detection in terms of silicate at the surface?
You mean like using this
Yeah.
tool? Yeah, certainly. I mean, I know there are.
That's more of a biology question, but
Yeah.
I just thought that was
That's
interesting.
okay. Yeah, I think, I mean, and in their paper, I think having some, I think they did have some discrete samples that they went out and collected, but it wasn't like a continuous time series from that period. So, yeah, as you said, like having those kind of real-time data would be really useful, and also useful for, I know there's some harmful algal bloom models out there, predictive models, and so including these types of information in those models would be really useful. Any questions on Zoom? Or?
I see one, but I don't understand the question. Yeah, I think something's missing from the question. You're only getting like wonderful, awesome, yay, and some French stuff that I understand.
So, unless there is no more questions, I think we can give Marine another round of applause. There is one more question. It came. So that's a question from Chris Measures back in Hawaii. He said that he noticed quite a bit of variation in the absorbance of a blank signal in one of your slides. I was wondering if you think that is beyond expected variation. Tough question.
Yeah, I think, well, there's a lot of things that can contribute to a blank signal, especially in seawater, because there's a lot of ions in seawater that might interfere with the signal of my blank sample. So if I'm running a blank that's just pure low nutrient seawater, I tend to see a slightly higher blank. And so that can be a little bit deceiving, but I mean, based on my, I guess, observations, the differences in blank that I see between Millicu and seawater were never more than like 0.01 absorbance units. So, yeah.
Yeah, I think it was referring to like the Fiona slide where you show the absorbance of the blank as a function of time. But yeah, as you said,
yeah.
I think if you look at the scale of the absorbance values on that, it looked huge, but if you look at the scales, it goes from 0.03 to minus 0.03 or something like
Right,
that.
right, yeah. Yeah, if you look at it, you're like, well, your blanks are all over the place, but yeah, that's kind of inevitable when you're working with such small numbers. So.
All right. Well, thank you all, especially those online and here.